Welcome to the Explainer. Today, we're zooming way in, down into the magnificent microscopic metropolis that exists inside all of us. We're talking about the cell. We're going to break down this fundamental unit of life that literally makes up every single living thing on the planet.
So, let's start with kind of a fun question. What do you actually share with a mushroom, a towering ostrich, or, a tiny invisible bacterium? Well, despite our pretty obvious differences, we are all constructed from the exact same microscopic building blocks. Whether you're a single-celled organism or a highly complex human, life fundamentally starts right at this exact point.
Here's our quick roadmap for today's microscopic descent. We'll hit life's building blocks, discovering the microscopic world, the anatomy of a cell, the cell boundary, and we'll wrap it all up with cellular transport and osmosis.
All right, jumping right into part 1, life's building blocks.
Think of this whole thing using a brick-by-brick analogy. Just like bricks make a wall, walls make a room, and rooms make a massive building, your body follows a very similar architectural blueprint. You start with a single cell. When a group of those cells get together to do a specific job, they form a tissue. Group those tissues together, you get an organ, like your heart or your lungs. When multiple organs team up, like your stomach and intestines, they form an organ system. And when you stack all those systems together, boom, you have an organism. You have you. When we study these fundamental units, we call it cytology. It's the field that proves literally every single function in your body, from breathing in oxygen, to digesting that lunch you just had, to fighting off a virus. It all starts right here at the cellular level. There is absolutely no escaping it. If you want to understand life, you've got to understand the cell.
Moving on to part 2, let's talk about discovering the microscopic world.
This whole microscopic exploration actually kicked off way back in 1665. A brilliant scientist named Robert Hooke designed his own microscope and looked at a thin slice of cork, tree bark. He saw these tiny, empty compartments that reminded him of small rooms, which is why he coined the term cells. But there was a catch. The cork cells he saw were totally dead. It wasn't until a few years later, in 1674, that Antony van Leeuwenhoek built a much better microscope and became the very first person to observe living cells actually moving around.
So a super crucial, really easy mental trick to remember this. L is for living, and L is for Leeuwenhoek. Hooke might have given them their name, but Leeuwenhoek actually watched them live and breathe, which basically earned him the title of the father of microbiology.
To actually see these living cells, microscopes have to bypass what's called the human eye's limit of resolution. For us humans, that limit is about 1 millimeter. Basically, if two tiny points are closer together than a single millimeter, our eyes just kind of blur them together into one single dot. Microscopes use lenses to artificially expand that distance, giving us the resolution power to peer into a world that would otherwise be completely invisible to us.
Let's keep zooming in with part 3, the anatomy of a cell.
Now, not all of these microscopic bricks are the exact same size. In fact, cells range drastically in scale. Get this, the largest living cell in the world is actually an ostrich egg. You definitely don't need a microscope for that one. On the complete opposite end of the spectrum, the absolute smallest cells are these tiny bacteria called mycoplasma. And if we look at length rather than volume, a human neuron, the nerve cells sending signals down your spinal cord, can actually be up to a full meter long.
But regardless of their size, inside almost every single cell is a bustling city with distinct, highly organized districts. You've got the plasma membrane acting as the strict border wall. You have the nucleus, which is your heavily guarded control center holding your precious DNA. Filling up the space is the cytoplasm, this jelly-like landscape where everything happens. And scattered all throughout are organelles, these specialized factories that do the heavy lifting, like mitochondria generating energy for the entire city.
Okay, let's take a closer look at that border wall in part 4, the cell boundary.
For a really long time, scientists weren't exactly sure how this wall worked. Then, in 1972, Singer and Nicholson proposed the fluid mosaic model, and it completely changed how we view this boundary. Turns out, the membrane isn't some hard, solid, impenetrable shell. Instead, it's this flexible, moving, fluid sea of lipids. And floating within this fluid sea are proteins that act as actual gatekeepers, deciding exactly what gets to come in and what has to stay out.
The lipids forming this sea, they're called phospholipids, actually have a bit of a split personality. They have a head that is hydrophilic, meaning it absolutely loves water. But they also have a tail that is hydrophobic, meaning it is terrified of water and constantly repels it. Because of this, they arrange themselves into a double layer, safely hiding their water-fearing tails on the inside, away from the fluid environments inside and outside the cell.
There's a really fascinating comparison to make here between plant and animal boundaries. Plant cells have an additional, highly rigid cell wall wrapped around their plasma membrane. Why? Well, because a plant can't exactly run away from a storm, right? It needs that rigid cellulose wall to stand tall against wind and rain. Animal cells, on the other hand, don't have this rigid wall. Our cells remain flexible, which is exactly what allows us humans to bend, stretch, walk, and move around freely.
And that brings us to our final section, Part 5, Cellular Transport and Osmosis.
Getting things across that cellular border requires different strategies. When things like oxygen naturally flow from a highly crowded, concentrated area to a less crowded area, it's called passive transport. It's kind of like coasting downhill on a bike. It requires absolutely zero energy from the cell. But if the cell needs to push a substance the opposite way, from an area of low concentration up into a highly crowded area, it has to use active transport. That's like pedaling uphill, and it requires the cell to actively spend its hard-earned energy.
Now, there's a very specific type of passive transport that gets its own special name. Whenever you hear the word osmosis, your mind should instantly jump to one thing: water. Osmosis is simply the movement of water navigating across those cellular gatekeepers. It moves from an area where there's a lot of water to an area where there's less, basically just desperately trying to find balance.
Because of osmosis, the environment completely dictates the cell's ultimate fate. If you drop a cell into a highly salty, hypertonic environment, the outside environment sucks the water right out of the cell, causing it to shrink. If you drop it into a highly watery, hypotonic environment, water rushes into the cell, pumping it up so it swells. And in an isotonic environment, everything is perfectly equal, so the cell shape stays exactly the same. An easy way to lock this into your memory is this great visual trick. Hypertonic has an R, which makes the cell shrink. Hypotonic has an O, which means it gets perfectly round and swollen. Works every time.
Let's animate this concept mentally with a really fascinating process called plasmolysis. Imagine taking one of those rigid plant cells we talked about earlier and dropping it directly into a very salty, hypertonic pool.
What happens next is incredible. Because the environment is super salty, osmosis dictates that the water must rush out of the cell. As that water escapes, the flexible inner plasma membrane starts to collapse inwards. But remember, plant cells have that extra armor. That sturdy, rigid outer cell wall absolutely refuses to move. It stands its ground, perfectly maintaining its rectangular shape.
Finally, the inner plasma membrane and all the cytoplasm detach completely from the outer wall, shrinking down into a tiny, shriveled ball sitting inside this large, empty, rigid box. This just brilliantly illustrates the extreme difference between the flexible membrane and the rigid wall. And honestly, it leaves us to wonder, if all this drama is happening inside just one cell, what other magnificent, microscopic events are unfolding right now inside your 3 trillion cells? Keep wondering, keep exploring, and I'll catch you on the next explainer.